All right, so today, this is going to be chapter 15, part 2. And as we've already discussed before I hit the record button, there is a set of PowerPoints for chapter 15, part 2, chapter 15, part 2. They are probably the PowerPoints from the 5th edition of the book. So some of the figure numbers might not exactly match. Again, with the note outline, they'll match with the note outline that you have, but the figure numbers might be off by 1 from your actual ebook that you have because it's a new edition from when we used it in the spring and summer. So today, this lecture, we're going to, if you remember from Monday when we were talking about the specific response or the non-innate immunity or specific immunity. And we had already discussed last time all about sort of B cells and particularly T-independent B cell activation, clonal selection theory. And today, we're going to spend our time talking about what we used to call the cell-mediated response. And it's called that because it's cells talking to other cells, so it gets rather complicated. So this is primarily T cells, but we're also going to see that some of these T cells and some intermediate cells, actually some of the B cells, also talk to the T cells. So we are going to see that some of the interconnections between what we used to call the humoral response and the cell-mediated response. So let's take a look here at starting with T cells and where they arise from. Similar to B cells, T cells, they're precursor cells, they're immature versions, come from the bone marrow and the fetal liver. And then they do something that B cells do not. T cells go to a place in your body, to an organ. It's kind of like this shaped, I think. They go to your thymus. So they don't go directly to the lymph like the B cells did after they came out of the bone marrow and encountered antigen and went to the lymph. T cells have a stop-off place at the thymus, and that is why they're called T cells, because that stands for thymocytes. And what happens to them in this thymus is they are selected, or they are, we used to call them, they're educated, in quotes there. And what we mean by educated is that if a T cell has sort of an incorrect receptor, meaning a receptor that is specific for self, an antigen that normally should not be allowed out into the lymph, they undergo apoptosis, which it stands for programmed cell death. So any T cells that have a T cell receptor, and that'll be more clear in a minute, that is specific for class 1 or class 2 MHC binding, as well as an antigen, a protein that is similar or is a protein of your own body, a non-foreign antigen, a non-foreign protein, I should say, they are eliminated by, again, by programmed cell death, apoptosis. After they travel through the thymus and they are selected, then those T cells leave the thymus and they go out to the lymph, and they're going to travel around to lymph nodes, and they're primarily going to be activated in lymph nodes. And that's when we would consider them mature T cells. And we are going to speak about 2 sets of mature T cells, the cytotoxic T cell and the T helper cell. And I realize your book has the helper cells divided into helper 1 and helper 2. They also have the T regulatory cells on there. I'll slap those on here at the bottom, T reg for regulatory T cells. Primarily, like I mentioned, we're going to focus on T helper 1 activation. Let's see, I think that's T helper 1. I can't read it without my glasses. Yeah, just a T helper cell there. I don't know what that stands for. And then, of course, we're also going to get T memory cells. Go to immunology, learn more about it. right? There are other subsets of T cells that have more specific functions, but in this class, for some of you, it will be the first introduction to the immune system. I'm trying to keep it as simple as possible. So let's talk about this activation, how these cells get activated, these T cells. We're going to start with the cytotoxic, and then we'll talk about the helper cells. And before we can speak to how they're activated, I would remind these do not produce immunoglobulin. They're going to produce chemicals that we in general call cytokines, but more specifically, you might see them called lymphokines. And most of them have been given a designation that we refer to them as interleukins. So these are special group of cytokines that help leukocytes speak to each other. They all have a designation of capital I, capital L, and then a number after them. And most recently, when I looked, which was a couple weeks ago, I think there are at least 33 of them that have been identified. We're going to mention maybe 3 or 4. Okay. All right. Now let's take a look at some of the features of the surfaces of the cells, immune and non-immune, in your body that help with recognition of a cell that is damaged or is infected with a virus, or actually has taken in foreign antigen and processed it and put it out on the surface. So there's sort of 2 sets of protein molecules that we need to talk about. And if this is the first section of your handout, the first set that we need to mention are what we call the MHC, and that stands for the major histocompatibility complex. Ooh, it's a long word. Mobility complex. And this was discovered in mice, and it's called because it was discovered and described when scientists were trying to transplant cells from one mouse to another. So that's why it says histocompatibility, the compatibility between cells of same species, but different individuals in that species. There are 3 major groups of MHC. We designate them as classes. And so they're numbered with Roman numerals. So there's class 1 MHC, Roman numeral 1, class 2 MHC, and there's also class 3 MHC. Now, this isn't it. There are more. There's also a minor histocompatibility complex, but we're not going to discuss that. I did put on your paper, on your handout, and I do want you to be aware that in humans, you will often see the MHC referred to as the HLA for the human leukocyte antigens. All right, so this is the same MHC. We title it HLA in humans. Okay, we're going to call it MHC in our studies, and your textbook does as well for the whole chapter. All right, so let's look at these molecules a little bit. So first off, class 1 MHC, this is a molecule that is on all your nucleated cells. All your nucleated cells. This is the main marker of self. All right. Each one of us has an individual class 1 MHC. It's going to be slightly different from our siblings, and of course, very different from our parents. And the less related we are, the less likely that class 1 MHC is going to match. All right, and the picture that they show in your book for what class 1 MHC looks like, I'm going to redraw a brief version of it because I can't access the PowerPoints at the same time here. So if this is the phospholipid bilayer of a cell, of a human cell, right, cell membrane, you'll notice that the class 1 MHC, your book draws it in blue and it sticks out. It has kind of this protein stick, and then they draw it as 1, 2, 3 blue dots, blue circles, and then a yellow one that sticks out to the side. Okay, so that's class 1 MHC. And again, you would find that on the surface of all your nucleated cells. So that means it's not on red blood cells, right? And it's there in more than one spot. So if I were to draw a whole cell, for example, with the class 1 MHC, let's see, I'm just going to draw it over here. All right, so if this was, I don't know, let's make it like a skinnier cell, like a skin cell or something, right? Like a epithelial cell. If this was like an epithelial cell and they're kind of squarish, right? There's the nucleus, the nucleolus. This class 1 MHC there would be multiples of versions of it all over this cell, especially in the areas where it contacted other cells and or where it was exposed to extracellular fluid, right, to interstitial fluid, right? So this little human cell would have multiple copies of class 1 MHC all over it, especially in the places where it would be likely to encounter other cells or fluid going by. Now, in addition to that, excuse me, in addition to that, cells in the immune system have class 2 MHC on their surface. And these cells would be macrophages. This is a shorthand for macrophage. Dendritic cells, B cells. These are the main ones that would have these on. And some T cells for talking to each other. Okay. Now, the class 2 MHC looks a little different because its function is different. Excuse me. Class 1 is just to say, hey, I belong to you. I'm part of your body. Class 2 MHC is more important in activating those T helper cells. So it's something that the cell needs to say, hey, I'm part of your body, and look what I have. I have this foreign antigen that I'd like to show you. And then if you are so inclined, you can make a response to that antigen. You can produce immunoglobulin or turn into a plasma cell if you're a B cell, or you can produce some of these cytokines or lymphokines. So the class 2 MHC looks different, and I'm going to erase my class 1 here so that I have space to draw the cell again. The class 2 MHC, phospholipid bilayer, right? Phosphate heads, lipid tails, hydrophobic, hydrophilic, right? The class 2 MHC, your book's kind of drawing it in brown, so I'll put it in red here because it's the closest thing I have. These are the protein tails that are embedded in the phospholipid bilayer, and then the physical structure of the class 2 MHC kind of looks like this. Kind of like that. Okay. And you'll notice in your picture, if you're following along in your textbook, page 415, figure 15.3, you'll notice that these cells, these B cells, dendritic cells, macrophages, in addition to the class 2 MHC on their surface, they're also going to have class 1 MHC because, hey, they belong to us. They're part of our body. Right? A particular feature that we need to recognize about this class 2 MHC is this area in here. That's the antigen binding site. So similar, this is where a bit of antigen is going to be placed, primarily to present or to show to T cells, as I said, hey, I have this. and it's not self, can you mount some sort of a response to it? Right. So this is this red one. This is class 2 MHC. And again, same if I drew a cell, but I am going to make it a round cell this time because it's probably going to be a B cell. This B cell would have class 1 MHC on it to tell the body, hey, I am self. And it would also have some of this class 2 MHC sticking out. I'm just going to simplify the drawing to say, hey, I have not only am I self, but that looks a little too much like antibody. Sorry. Make it look like little antenna or something. I also have a place to bind antigen. So this, for example, might be a B cell or a macrophage. If it's a macrophage, it's a very busy surface because remember, also on the surface of macrophages, there's receptors for FC portion of immunoglobulin. And we're going to find out they have receptors for other molecules too. I should draw them in different colors if I can. Right. Triangle, circle, square. Sorry, it's about all I can draw. But anyhow, so the surface of these cells is sort of jam-packed. It's not just a single item that's sticking out there. And we are not going to do any more discussion about class 3 MHC, except to say that these molecules are mostly involved in complement. Now, as if this isn't complicated enough, there are another group of molecules that we refer to as, abbreviate them as capital C, capital D, and that stands for clusters of differentiation. This helps us identify the immature molecules, the immature cells, the precursor cells. For example, if we were to list, you might remember the other day I mentioned CD28. That's a marker for a pre or an immature B cell. We're going to see CD3. This is a marker or a protein that is expressed on all T cells. It is usually very close to the T cell receptor. So I'm going to put near TCR for T cell receptor. That's the part that's going to bind antigen, and it's also going to bind to that class 2 MHC that we mentioned. And then we also have some other common ones that we discuss are CD4. This is present on predominantly TH or T helper cells. And CD8, which is predominantly present on cytotoxic T cells. Notice I'm saying predominantly because there are some quirky cells that might be CD8 positive but have cytotoxic, or CD4 positive but might have cytotoxic abilities, but we're not going to discuss those now. And usually people designate a cell if it has one of these markers on it. For example, people just write the CD28 positive, right? CD3 positive, like that, to indicate that particular protein molecule is present. So these are also proteins. And they're markers. We think of them as markers, clusters to help us as scientists figure out what group of immune system cells do you belong to, right? And again, there are many, many, many of these complementary clusters of differentiation. Your book says there are close to 400 have been described, and I've mentioned 4. Okay, so you can learn more about those when you travel on to your next class. Now, one more thing that I would add, and this is sort of in letter B here of the lecture notes, the antigen binding receptors and other cells of the immune system / markers, list the markers present on B cells, the markers present on T cells. I would remind you that the T cells, which we're discussing today, right, the T cell receptor T cell receptor is similar. You can think of it, it's kind of similar to immunoglobulin. It has two chains. It has an antigen binding site. And like I mentioned, all T cells are going to have some sort of a T cell receptor, plus they're always going to have CD3 on their surface. And that CD3 is really important because nestled in that CD3 is going to either be CD4 or CD8. And these are going to sort of be co-binders to the antigen. They're going to co-bind to the antigen. So the T cell receptor has a place for antigen binding, but the T cells themselves don't process the antigen. They don't chop it up and put it into pieces and put it out on the surface of their own cell. They need someone to show it to them. So their T cell receptor is going to bind antigen, and this CD4 or CD8, they're going to bind to antigen plus, they're going to reach around and bind to, actually I should change that, not co-bind, let's say this, let's say bind to either class 1 or class 2 MHC. All right, so now let's take a little bit of a look at the cytotoxic T cell response. And again, in your handout, on your handout, it says figure 1511, and I believe it is actually figure 15.12. So I'm going to change that on my handout as well. And notice I've indicated it's the bottom panel. So I'm going to just redraw that a little bit for you here while we talk about what happens with the cytotoxic T cell. I'm choosing to do the cytotoxic T cell first because I think for your first exposure to how a T cell is activated, this one's a little bit simpler. Okay. So cytotoxic T cells, which we indicate like that for a subscript of a small c for cytotoxic, they, on their surface, they are going to have a T cell receptor, which of course is going to have some CD3 associated with it. And that T cell receptor, they're also going to have, they're most likely going to be CD8 positive. So what we're going to see is the CD8 and the T cell receptor together are going to bind to antigen, and it's a piece of antigen, not the whole thing, like with a B cell. Bad drawing there. It's going a piece of antigen, piece of processed antigen. And then the CD8 portion is going to bind to class 1 MHC on another cell. Right? So this cell here that has the antigen and the piece of antigen and class 1 MHC, this cell we refer to as a target cell. And it could be just any old cell in your body. Most of the time, oftentimes, T cells, cytotoxic T cells, are stimulated by cells that have been previously infected with a virus. So if I were to try to draw this for you sort of as it's happening, usually let's draw the target cell as a different shape. Okay, so let's make the target cell, again, one of those kind of rectangular general cells. Maybe it's a liver cell. Maybe it's infected with hepatitis virus. So over here, I'm going to draw, let's make him red. So over here, I'm going to draw the virus with its spikes on its surface. There's some DNA in the virus. right? So this is the virus that's getting into the target cell here. And in this case, like I said, I'm saying this is the liver cell. It could be any cell of your body, right? And this, let's say, is hepatitis virus. So the hepatitis virus is getting into the cell, and then remember what viruses do. They take over the machinery of the host cell, and they start to make virus particles, right? Their job, they're focused on making more virus. While they're doing that, using your machinery inside your body, inside your individual cells here, while they're making more virus, some of the virus particles are being put out on the surface of this cell, of the target cell. It's a little bit of the virus protein, right? I'll try to make them look round, being put out there. And remember, this is a regular old cell of your body, so it's going to have class 1 MHC out on the surface. So these get put together very close to each other, both virus particle and class 1 MHC. So next to all of these, I'm going to draw some class 1 MHC. Again, I'll try to label that. All right, so this is class 1 MHC because this is just a regular old cell. And this little red dot is viral protein, right, which is antigen. Now, along comes the cytotoxic T cell, which I'm also going to make rather large so I can draw all its pieces. This is our cytotoxic T cell. And the cytotoxic T cell, remember, is going to have a T cell receptor on its surface, which I think we were drawing those in green. Your book is putting them kind of as yellow here. So I'm going to draw this T cell receptor, which has a little spot for to bind antigen. Right? And remember, right next to that is some CD8, which I'll draw as black here, like a black lollipop. How about that? So that's supposed to be the CD8. The green thing is the T cell receptor. Now, when this combination engages, so when or if the cytotoxic T cell receptor binds to class 1 MHC and antigen on the target cell. Right? So the T cell receptor being the T cell receptor plus CD8 in this case. And don't forget, we also have sets of CD3 on there that are actually holding the CD8 in place. So the yellow is CD3. It's always an association with the T cell receptor. And of course, remember, this particular T cell is not, it doesn't just have one TCR on it. It's going to have multiple TCRs and multiple CD8s and obviously multiple CD3s all over. I just drew two. All right, but back to when or if the cytotoxic T cell receptor, I should put and CD8 bind to class 1 MHC and antigen on the target cell, then something's going to happen. Then, right? If, then the cytotoxic T cell is going to release some pretty potent compounds. We call them perforins and granzymes. And they are pre-made, and they are inside of vesicles here. I'm going to make them blue and green. We should probably make them red and yellow because they're so dangerous. And the cell may have more than one package of them. Usually just draw one, but I'm putting a couple packages in there. They're going to release these perforins and granzymes, and what these compounds do is, and again, I'm just going to focus on the red ones, these perforins and granzymes are going to be released out here into the intercellular space and extracellular fluid, and they are going to go and literally perforate the cell membrane, and they are going to kill and eliminate this target cell. Which will stop the virus from being able to reproduce, particularly in this cell, and we're just going to completely get rid of it. This then, of course, is going to stimulate inflammation. So think back to the last chapter. Complement may show up here, right? As well as it's going to call in macrophages, part of that inflammation, that are going to then be able to stimulate the helper response. So it's all interconnected, right? All right, now I'm going to add another board for the T helper response, which is slightly different, but similar, right? So the T helper cell looks like this. And this helper cell is going to be CD4 positive. Page here. I guess, yeah, they're showing it here on, I would look at figure 1510. So this T helper cell on its surface, and there's a nice blow up in your book. I'm going to try to redraw some of it off to the side here, is going to have a T cell receptor, right, with a space to bind antigen. It's also going to have CD3 on its surface, which I'm making in yellow. I realize in your book it's blue. And embedded in that CD3 is going to be, and oh yeah, I'll make it red, is going to be CD4 molecules, which I'm going to draw as a triangle. Remember my other lollipop for CD8 looked like a lollipop. So this is the CD4 portion. Right. And then there is another molecule, particularly attached to a helper cell that is similar to the B cell CD28. It is CD28. So I'm going to draw that out here. Looks like it needs to be on the opposite side of the CD4. So I'm going to put that molecule out here. Let's make that one blue. I can get rid of that. It's kind of this long stick, again, another lollipop, CD28. Okay. Now, this T helper cell, it does not produce immunoglobulin, and it does not produce perforins and granzymes. It's a helper cell. It sends out molecules that are going to help with inflammation, help with phagocytosis, help with calling in cytotoxic T cells. So its function is to help, right? Now, it is activated or stimulated when, and this is supposed to be a macrophage here. So an activated monocyte that has become a macrophage, with an O like through that, that's an abbreviation for macrophage. Right. And what this macrophage has done, and let's use a bacteria this time for our antigen. So along comes our bacteria over here that has blue triangles. We'll make them circles, blue circles on the surface. Right. That's going to get ingested, phagocytosis. Right. the cell is going to break apart. The phagosome is going to fuse with the lysosome, right? And I'll make that one red because lysosome has all the enzymes in it. We're going to get that phagolysosome that is going to produce bits and pieces of my bacteria, right? Some of those bits and pieces of the bacteria are going to be placed on the surface of our macrophage, bound to class 2 MHC. So another vesicle actually fuses with this one that is going to have class 2 MHC. I'm trying to draw this that it looks sort of like the picture in your book. Class 2 MHC, remember, kind of looks like those two little things like that. And this class 2 MHC is going to present the antigen, which I'll keep it blue, circle. So the antigen's bigger than this, right? It's going to present the antigen because it's going to bind into the antigen binding site of the T cell receptor. The CD4 is going to bind to the outside of this class 2 MHC. We consider it like a handshake and something we don't do anymore, a handshake and maybe holding on to the person or giving them a hug as well. So this is going to bind here. So we say that the T helper cell needs to see or recognize antigen in the context of self, antigen in the context of CD4. In the context of class 2 MHC. And then the third portion here, this is still relatively new, is this portion, this on this cell, on the macrophage or the antigen presenting cell, there's another cluster of differentiation that is CD80. And we need all of these things to bind in order for the T helper cell to get activated. So we need the T cell receptor to bind to antigen. We need the CD4 on the surface of the T cell, the helper T cell, to bind to class 2 MHC. And we need this CD28, CD80 interaction. So I'll just put this here to remind class 2 MHC. Yeah. Okay. Then it becomes a very similar scenario. If, but one more thing, we refer to this cell, this macrophage, and it could be other things besides a macrophage. It could be a dendritic cell, or B cells are actually very good at this as well. We call them antigen processing cells, or APCs. Okay. And again, it could be a macrophage. B cells are particularly good at it. Dendritic cells are also particularly good at it. And then they show you a picture. Now, if you speed ahead to figure 1512, they show you the results of activating this T helper cell. Okay, so similar to how I had to the right-hand side of if the cytotoxic T cell was stimulated, if the CD4 positive T helper cell binds antigen plus class 2 MHC, and also this CD28, CD80, then something's going to happen. Then there are possibilities. There is going to be primarily release of IL-2, interferon, and IL-4. And they all have different functions, but let's just say that so then we're going to get IL-2 released. And if this is a T helper cell 1, this IL-2 that is released is going to stimulate. It does a couple of things. It stimulates proliferation and differentiation of T helper 1 cells. The differentiation is going to be into a memory cell. It also, IL-2, also, it used to be called B cell growth and differentiation factor. You can see why they switched it to IL-2. But why I tell you that is because it helps B cells grow, basically mitosis, differentiate, and further stimulates the production of immunoglobulin, sort of indirectly, right? And IL-2 can also, so I'll put this as the third thing, it can also stimulate the growth and differentiation of cytotoxic T cells. So it can amplify the activation cycle that we talked about in the last panel. Right, so we're also, so when these bind, like I said, primarily the big ones to focus on, this is going to, the T helper cell is going to release the IL-2. However, the macrophage can also release and bind IL-2 as well as IL-4 and gamma interferon. Okay, I think that's it as far as I'm concerned about what you need to know about T cell activation. Panel 1513, right, describes particularly how B cells as antigen processing cells interact with T helper cells, which is just an amplification of what we've already talked about, but this not being a macrophage, but it being a B cell. So if the antigen presenting cell was a B cell, we get a little bit of a different combination of presentation. This is missing. The CD28 and 80 is not a part of this, but the T cell receptor is, of course, as well as the class 2 is. So I'm going to just draw that real fast with my CD4 and that binding to class 2 over here, like that. And of course, still this would have CD3 around it, but we don't have this portion here. That's what's missing, the CD28 and the CD80. That's only if it's a macrophage. This then is going to activate other B cells to become memory cells and plasma cells. And I think last thing I did not talk about, I didn't mention IL-10, but that's important for the T regulatory response. That's important for helping shut the immune response off, I believe, the IL-10. Let's see, regulatory, it just doesn't say in your book. And then we also have the T helper cell 17, which promotes inflammation, and you can see that on the panel, figure 1512. Now, last but not least, we do want to mention, this isn't really last but not least, we do want to mention primary and secondary immunity, because this is, of course, the primary immune response and the secondary immune response, because this is important in vaccination. And now that we have the release of information that a company in Germany has produced a vaccine and has given people 2 doses, they will be measuring both the primary and secondary immune response of the people that have received the vaccine. They'll be measuring how much antibody they're making, basically, to COVID virus particles. So let's take a look. If you take a look at figure 1518, basically, we just want to say that this primary response is predominantly a high amount of IgM and a relatively low amount of IgG. The primary response takes about 7 to 14 days, depending on the health of the person. And then you wait for a period. It's usually a month or maybe another 14 days until this primary response has decreased to almost undetectable. Then you challenge, we say, or give a second dose. And then you get a secondary response. And that secondary response, the characteristic of it is a little bit different. First of all, almost all of the antibody or immunoglobulin is IgG. The amount of IgG in this secondary response is much greater than either the IgM or the IgG in the primary response. And it occurs much shorter time period. So this we usually say is 3 to 5 days. Oops, it's not a day, is it? 3 to 5 days to get that secondary response. This is important because this is how vaccination works, right? The idea is your primary response is going to be your exposure to the vaccine or your first exposure to the antigen, and then your secondary response, which is going to occur much faster, much higher immunoglobulin. Notice it's mostly IgG, which if you think about from our last lecture on Monday, is much more versatile immunoglobulin, does a lot more different things inside the body, and it's a much faster time, right? This would be when you actually encounter the antigen, right? And the hope is that if you've made a strong primary response, your secondary response will basically prevent you from getting very ill or ill at all. Which brings us to a little discussion about vaccines. And I did a list there, a checklist for effectiveness of vaccines. Oh wait, we forgot to talk about the different types of immunity. Sorry, I was getting ahead of myself here. All right, immunity, we call all of this immunity is acquired immunity. And it basically falls into two categories: naturally acquired or artificially acquired. And that's like it sounds, right? Naturally acquired, you just get sick, or you were exposed. Artificially acquired, you got a vaccine, right? And then this falls into two categories: active and passive. So, and it repeats. So naturally acquired passive immunity is when you are a fetus and you get immunoglobulin G from your mom across the placenta, and when you are a neonate, which means right after you're born, and you get some IgA from your mom from if you're breastfed, if you get breast milk. Naturally acquired active immunity, you get sick, right? You encounter antigen in the environment, and you either get sick or you produce a primary immune response. Artificially acquired passive is, well, let's do artificially acquired active first. This is you are vaccinated and you produce a response, but it's a forced response. It's not naturally occurring. You're not getting the whole organism. And artificially acquired passive, this is when you receive, it's usually an immunoglobulin cocktail in the form of a serum transfusion that is immunoglobulin from people who've already recovered from this disease. Okay. Or it's a prefabbed monoclonal antibody. So, but it's usually pooled IgG. I think there's two L's there. Pooled IgG from people who've already recovered from this disease. And the idea is by giving you a large dose of it, it'll be able to help your body quickly neutralize the virus or the toxin rather than waiting for you to form a naturally acquired active response, because you might not have time. You might die before this gets finished, right? If we have to wait 7 to 10 days or 7 to 14 days. All right, which brings us to vaccination or vaccines. And I know we've been running long here, but just very quickly, right? There's a little checklist there about the requirements for an effective vaccine, just like we had requirements for an effective antibiotic, or chemotherapeutic agent from chapter 12. It's pretty common sense, like I mentioned in the handout. I'm not going to go over it specifically, but you should look at it, and I'm going to include it on the exam. The categories of vaccines, the oldest types of vaccines are whole cell, bacteria or virus. These are more effective, but they are more likely to cause an allergic response. So whole cell, we mean killed or inactivated. So the most common way to kill or inactivate is using heat, or you can use chemicals. What is referred to as live attenuated. It's kind of an oxymoron because attenuated means, you know, it's still alive, but whatever. I'm going to spell it wrong. Attenuated. This means partially killed or partially inactivated, right? There's still a little bit of live virus or live bacteria, but so small of an amount that it's not enough to cause an infection, to cause illness. And again, benefits and risks, they're listed in the purple table there, right? The advantages of a live prep are as follows. Usually you get a stronger response. The response is more long lasting. They usually require fewer, because of that, you need fewer booster shots, and they are really effective at getting a strong cell immediate immunity, or getting those T cells activated, which is going to be really important for quick activation of the B cells later. Now, the disadvantages are they're difficult to store. The live attenuated in particular can be transmitted to an uninoculated or an unprotected person. So for example, if that's what happened with polio virus, the oral version of polio was the oral version of the polio vaccine, right? Children who got that were shedding virus in their feces, and their younger siblings could pick that up who had not yet been vaccinated, right? So that's what we mean by it can make other people sick. What's more common is what we call a subunit vaccine. This has less side effects as far as allergic response or a non-specific inflammatory response. However, these cannot cause illness at all because it's only part of the organism, but they are not usually as effective. So a toxoid would be an example of a subunit vaccine, right? Like tetanus toxoid. And you don't get any or very little of the whole real organism. They can be biotechnology derived, so there's no actual organism, and you get the toxoid from that. But they require much more frequent, like every 10 years or so, right? They require more booster because they don't make as strong, you don't make as strong of an immune response. Now, the wave of the future, which is what I believe I heard yesterday, the vaccine that's being that's already been tested on 40,000 people it's double-blind study. So 40,000 people, 20,000 of them received placebo, 20,000 received vaccine, and they have 2 doses of the vaccine because it does require a booster. they're getting I believe it's a messenger RNA vaccine. They didn't talk much about the science on the radio and PR yesterday, but they talked more about the administering and how quickly it could be made into enough to distribute to the wide population in the United States or the world. It's a messenger RNA vaccine, which is it's an interfering RNA vaccine. So it's given, and what this does is it prevents the coronavirus from being able to reproduce. So it blocks its reproduction, because it essentially makes double-stranded RNA, and then that can't be read by either the host cell, and the virus then can't be multiplied, right? So think back to transcription and translation. The biggest challenge with the vaccine, from what I heard, is that it has to be stored at like -80, -90 degrees centigrade. So it's going to be difficult to administer as far as all the places getting the correct storage facility so they can actually maintain the vaccine at a cold enough temperature that it will be effective. However, bright spot. There's also a list of recommended immunization schedules for both children and adults. You should take a look at it. It's on page 438. And it gives you some sense of why do we give so many vaccines to children? It's because they have bigger thymuses. So your thymus, right, is large when you are born, and as you age, it decreases in size significantly. So you are less likely to be able to develop a strong cell-mediated response the smaller your thymus is. So the bigger the thymus, the better. All right. Does anybody, I'm going to